Bismillah Rahman Rahim, O level biology, new component. So, there are some new questions on the paper 2 which some students have asked me to talk about. The topics on which the questions are going to be discussed is lactose free milk, pectinase, and biological washing powders. Please use headphones. You all complain of the volume being very low or something like that, but I have researched all this issue a lot, and please, everybody, please use headphones, so that will be more helpful for you all. Now, when you look at the first question, it's a mixture of this question going on to a little bit more about lactose. So I have researched the whole question, and I want to talk about the whole question for you all. Milk is sometimes referred to as a complete food because it contains all the nutrients that a young mammal requires. Table 1.1 shows the three nutrients that are contained in milk. Complete the table by stating one role of each nutrient in the body of a young mammal. Sometimes you want to do this question, so you can always type the first opening sentence of the question and you'll get to know which year's paper I've used or which resource have I used. So, what is the role in the body of proteins, lactose, and calcium? Now, basically, you can give me only 1 point, and that should be enough for the 3 marks that you have for this part of the question. So, proteins are needed for growth. Or if you didn't say that, you could have said making new cells, or you could have said growth and repair or replacement. And what does it do? Proteins are required because they will be digested to amino acids, and the amino acids are then broken down to make, like if you drink milk and it contains protein, what happens to the milk, the protein in the milk? The protein in the milk is digested to amino acids and they reach each and every cell of your body. So when they reach the bone marrow, maybe those amino acids are used to make hemoglobin. Maybe they reach the lymphocytes, they're used to make antibodies. Lymphocytes make antibodies. Maybe they reach the muscle cells and there those amino acids are used to make myoglobin. So there are a whole lot of, they reach the pancreas and they're used to make the protein hormone insulin. Please remember, all hormones are not proteins. Some hormones are proteins. There are some hormones which are steroidal, like estrogen and progesterone and testosterone. Those are steroidal hormones. So, what do we need for proteins for? We need them for growth. You can say growth and repair. You can just say growth. Anything was right. Very easy 1 mark. Then why do we need lactose? You see, what you have to understand is lactose is broken down to glucose. And this glucose is going to be respired to release energy, not produce energy. That would be wrong. If you said produce energy, that would be wrong. That's a reject. So that means a large number of students wrote that. And I keep on saying it, yet you will again write it in your paper. So the glucose is respired to release energy. So basically, why do we need lactose is that it provides energy, or you can say it provides energy. It is respired to release energy. So the word is release energy. Then what do we need the calcium for? The calcium is for strengthening the bones and the teeth. And it is required for blood clotting, it is required for muscle contraction, it is required for nerve impulse conduction. But it needs vitamin D. It is needed, calcium is required because vitamin D, for the vitamin D to function, because vitamin D and calcium both together are needed for the absorption and the deposition of calcium. So vitamin D is also important, needed for vitamin D to function. So protein, growth and repair, lactose broken down or digested to glucose, and glucose respired to release energy, and calcium needed to strengthen or make them stronger, the bones and the teeth, and for blood clotting and for muscle contraction. Now coming to the B part of the question. Protein digestion begins in the stomach of the human alimentary canal and is completed in the small intestine. Describe in detail how enzymes function to digest protein in the alimentary canal. So basically, how enzymes function. So you're not going to be talking about the mechanical digestion, you're only going to be talking about the chemical digestion. So in the chemical digestion, you know which enzymes you're going to be talking about. So enzymes are biological catalysts, they speed up reactions, they're specific, then they have an active site. where the substrate fits, and that is the complementary shape of the molecule. Then the enzyme substrate complex are formed. So it is actually they asking a question is how enzymes function. So it's a chapter on enzymes. Then enzymes lower the energy needed for reaction. Enzymes are unchanged. Enzymes carry out chemical digestion, break down large insoluble molecules into soluble molecules. Then proteins will be broken down into peptides, and then peptides into amino acid. Then we have a very important enzyme pepsin, which is active in the stomach. Then we have a very important enzyme trypsin in the small intestine. And in the stomach, the HCl will provide it the pH, which should be between 1 and 3. And then in the small intestine, we require a pH of 7 to 9. So that is alkaline. And that, of course, will denature the enzyme, and as the in the stomach, the HCl will denature the amylase, which is coming in from the mouth, which has been produced by the salivary glands. Then the temperature, optimum temperature, 37 degrees Celsius, where they're going to be more successful collisions because that's the body temperature. But of course, there are about 18 mark scheme points for a 6-mark question. So any 6 of those, and you got your 6 out of 6. So a very, very easy question. Describe in detail how enzymes function. So basically, how enzymes function, you had to give me the enzyme story, not just the story in the alimentary canal, but the whole thing about how enzymes can work. So in order to get your 6 marks. So please pause the video here and write these down so that then you know the exact wordings. And then, of course, when you write something and you read something, and if you read it aloud, you will remember it in your exams as well. So substrate fits into active site. The fact that we're talking about biological catalyst and the fact that we're saying it is specific. Specificity means that we're referring to the enzymes being specific. And the reason why, because they have a certain shape of the active site, which is a depression on the surface of the enzyme. Then substrate fits into active site, which is complementary shape to the substrate, enzyme substrate complex formed. We can also write ESC, enzyme substrate complex formed. Enzymes are unchanged. Enzymes lower the activation energy. Enzymes carry out chemical digestion, break down large insoluble to small soluble molecules, protein to peptides to amino acids, and then propepsin is active in the stomach, and trypsin in the small intestine. And we have to talk about the pH in the stomach is 1 to 3 and 7 to 9 in the small intestine, and the optimum temperature is 37 degrees Celsius, which is the body temperature. So lots of mark scheme points, but any 6 and you would have got your full marks. Now the C part of the question says some people are unable to digest lactose, which is the milk sugar, and have a condition known as lactose intolerance. Figure 1.1 shows what happens in the intestine of a person who is lactose intolerant if they eat food containing a lot of lactose. So from the stomach, you can see there is buildup of gas in the small intestine and in the large intestine. And bacteria produce hydrogen, methane, and carbon dioxide. Bacteria digest lactose to simple sugars, and there's water, and this is what is going to happen. So you can see there's a diagram showing lactose and the bacteria are digesting the lactose to simple sugars, and the bacteria produce hydrogen and methane and carbon dioxide. So the person feels very bloated, or he will say, I've got a lot of gas or something like that, to show that he's not very comfortable. So there is diarrhea and gas. So it's going to be diarrhea means frequent passage of watery feces. So he's going to be going to the toilet very often and passing watery feces. Explain why lactose is not absorbed by the small intestine. Well, because lactose is a large molecule. There's no enzyme. In these people, there's no enzyme to digest lactose or to break down lactose. And the lactose molecule is too large. It cannot pass through the cell membrane. And there is no carrier proteins for it. So the bacteria are going to use that as food and multiply and produce gas. So explain why lactose is not absorbed by the small intestine. So you can see the main reason why lactose is not absorbed. Why? Because why is cellulose not absorbed? cell wall is made up of cellulose, and that is the fiber in the small intestine, is because there is no enzyme for cellulose to be digested to glucose. Cellulose is made up of glucose. But human beings do not contain any enzyme, but cows and sheep, herbivores, contain actually bacteria living in their stomach which produce the enzyme cellulase. So explain why lactose is not absorbed is no enzymes to digest lactose. Lactose is a large molecule. It cannot pass through the cell membrane, and there is no carrier proteins for it. You see, for certain large molecules they can pass if there is a carrier protein for it. But of course, there is no carrier protein for it in the human cell membrane. Then part 2 of the question suggests the dangers to health of severe diarrhea if it is not treated for a long time. Yeah, there's going to be dehydration. Why? Because there's going to be a loss of water, loss of salts, loss of vitamins, loss of minerals. And this is going to result in decrease in the volume of blood. So when the volume of blood decreases, the blood pressure decreases. And if the water is lost, then there's going to be increase in the blood concentration, and there's going to be that is going to result in the tissue fluid, and that could affect the cells. Either the cells could, if the blood is too concentrated, then the water will move out of the cells and they will shrink, and those cells can die. So all of this is going to affect the long term, will affect a lot of the body cells being damaged. The dangers can be dehydration. Dehydration means that the person is passing so many frequent watery feces that he is going to lose a lot of water, and of course the water comes from the blood, and he's going to lose a lot of ions as well. So if he loses a lot of water, then what is going to decrease? The volume of blood will decrease. If the volume decreases, then the blood pressure decreases. That is why we are saying the blood pressure is going to decrease. And the blood will become very concentrated because it's losing water and some ions, not all the ions. So there's going to be an increase in the blood concentration. If this blood becomes too concentrated, then the tissue fluid becomes too concentrated. And this, of course, the cells can shrink and die. So all these are the effects of severe diarrhea. Then let's come on to the D part of the question. Hydrogen gas is produced by the bacteria that digest lactose in the large intestine. The gas is absorbed into the blood and excreted through the lungs. Lactose intolerance can be monitored by measuring the hydrogen gas content of the air a person breathes out. So what you have to understand is hydrogen gas produced by the bacteria which digest the lactose, then the gas is absorbed into the blood, and then it is going to be exhaled out. So the lactose intolerance can be monitored by measuring the hydrogen gas content of the exhaled air, of the air the person breathes out. Of the air the person breathes out. Understand that, that's exhaled air. People taking part in an investigation into lactose intolerance consumed the following milk products on different days. A. Untreated milk, normal milk. A. Untreated milk. B. Milk treated with lactase immediately before drinking. So this person did not have any lactose. Then milk treated with lactase 3 days before drinking. So 3 days before drinking, it was treated with lactase. And then the D part, C was the milk treated with lactase 3 days, and D was yogurt made by bacteria that digest the lactose in the milk. That's why the lactose is converted to lactic acid in the milk. So yogurt made by bacteria that digested the lactose in the milk. The hydrogen gas content of the air breathed out, which means exhaled air, was measured every hour for 5 hours following the ingestion of each milk product. The mean results are shown in figure 1.2. Okay, now I'm going to give you time to look at this graph, and I want you to pause the video here and then look at the graph, and then I'll read the questions on this graph. Now, as you look at the graph, you see it's at the hydrogen gas content. This is the hydrogen gas which we are measuring. The hydrogen gas breathed out in parts per million. So you can see the untreated milk A had the highest amount of hydrogen gas, which peaked at 3 hours. Then B was the one milk treated with the lactase immediately before drinking. Now that peaked as well, but peaked lesser, and the hydrogen gas was a little more than in A in the beginning, and then it peaked a little. So the hydrogen gas already breathed out in the beginning was more than it was in A, and it ended up even less in 5 hours. Then milk treated C Milk treated with lactase 3 days before drinking. That had very little hydrogen. Even in the beginning and even later on at 3 hours, it had the hydrogen was very little. And then it sort of increased surprisingly in the 3 to 5 hours. And then in D, yogurt made by bacteria. If you took yogurt, well, it remained less than 10, and a little bit up, so 10 parts per million, and then it remained low, less than 10 all the time. So the least amount was in the yogurt made by the bacteria, so less hydrogen was being produced. Then part one of the question explained why untreated milk was included in the investigation to compare. It's a control for comparison to see if there is any difference between the effects of treated milk and untreated milk. It is always you have to do a control. Control means something that you have to prove, you compare that, okay, we did this, but then when we didn't do this, what would happen? So a very simple way is that it was the control. Now, the answer to this was it was a control for comparison, or if you said to see if there's any difference between effects of treated milk and untreated milk. Now, some of you wrote it is a fair test. Now, that was wrong. Fair doesn't mean this. Fair means that you did nobody cheated. There was no cheating. There was no unfair means. That is not what you're trying to say. That's wrong English. It was to ensure that it's a fair test. So, I mean, how do we ensure that's a fair exam that you take? You're not allowed to take your mobiles. You're not allowed to take your Apple Watch with you. Your some exams don't allow even a calculator. Some exams allow a calculator. So all those things make it a fair test so that nobody's at an advantage or at a disadvantage. And part two of the question suggests why lactase might be added to milk. Why would we add lactase to milk? Lactase is going to digest the lactose, so that the molecules, then the glucose and the galactose, are small enough to be absorbed. So it will reduce the chances of diarrhea, or you can say it means that lactose intolerant people can consume milk. So lactase digests lactose, molecules then are small enough to be absorbed and reduces chances of diarrhea. So the person will not have diarrhea, will not get dehydrated, and will not have to be rushed into hospital and given drips and medicines to control the diarrhea. Part 3, use the results in figure 1.2 to explain why yogurt is the best milk product to people with lactose intolerance. So why is yogurt such a good idea? Looking at the graph, let's look at the graph again. Now when people had the yogurt, what was happened? Hydrogen is the lowest. The concentration of hydrogen was the lowest, or you can say the amount of hydrogen was the lowest. And the hydrogen concentration shows small fluctuations. They weren't very large fluctuations. They were very small fluctuations. Look, it's gone down a bit, then up a bit, then up, then it's like this. And the concentration not higher than 15 parts per million, between 9 and 15. And look at D and A and B and compare it with A, B, or like if you said with A, it was more than 50. And here it has remained at 3 hours. Here it is at 10, and A was nearly 50. So if you give me some comparative figures and data, this is called a comparative data quote between D and A or D and B or D and C. So concentration of hydrogen is the lowest. Concentration of hydrogen is the lowest for yogurt. And concentration of hydrogen shows small fluctuations not higher than 15 parts per million. ppm is parts per million. This is ppm, parts per million. And then whenever it says, results in figure, refer to a figure which is given. So this was the figure that was given to you, figure 1.2. Whenever it says that in the question, then you must give me a data code. A data code means that you have to give me something from the x-axis, which is 3 hours. At 3 hours, you could have said at 1 hour, 2 hour, anyone which you would want. Like I liked it at 3 hours. What was the figures? For A, it was 51 parts per million. For B, it was 42 parts per million. and for C 20, and for D it was 10 parts per million. So I've done this on the graph as well. I've shown you how I figured this out. I so just read off the graph all these points which came on this line, and then I've written those on the side. And then this is called a data quote. Data quote means you start off on the x-axis, you take anything on the x-axis, and then you read the corresponding readings on the y-axis, and this is called a data quote. You must do that whenever in the question it says reference results, use the results in figure flan flan flan, or figure 1.1 or 2.2 or 7.2 or whatever, to explain something. Question 3. Starch, glucose, and fructose are carbohydrates. Fructose syrup is used as a sweetening agent as an alternative to sucrose. The flowchart shows how fructose is prepared from maize starch. So you take the maize grains are crushed and the pH is adjusted, and the starch is released from the maize grains, and you add enzyme 1, and then it forms into glucose, and then you add enzyme 2, and it forms fructose. Please remember, glucose and fructose are isomers. Now it says name enzyme 1. Now, what is going to convert starch into glucose? It has to be amylase. Yeah, it's a little bit of a thing. You could say, Miss, amylase converts starch to maltose, and then maltase converts maltose to glucose. But then they've said, okay, glucose syrup. So the first thing that they asked you, name enzyme 1. So that is amylase. Then it says state why it is necessary to adjust the pH before an enzyme is added to the process. Why? pH is a factor that influences enzyme activity. And in order to provide the optimum pH. Why? Because extreme pH can denature enzymes. So the pH affects enzyme activity and it affects the rate of enzyme activity. So in order to give the optimum pH, we need to add either an acid or an alkali to adjust the pH. And then extreme pH could denature enzymes. So the whole story would stop because the enzyme just won't work. Now, maize grains contain protease enzymes. With reference to the processes shown in figure 3.1 suggests why it is important that these enzymes do not contaminate the glucose syrup. Why? Because the protease would break down the enzymes, the enzyme 1 and the enzyme 2. So, maize grains with reference show why it is important that these enzymes do not contaminate. Because what I'm saying, maize grains contain their own protease enzymes. So, we don't want the protease would break down enzymes, would break down enzyme 2 which we added, would break down enzymes. Or you could have said enzyme 2. Then coming to part C of the question, the formation of fructose syrup from glucose syrup is carried out at a temperature of 60 degrees Celsius. Suggest an important property of enzyme 2 that allows it to be used at temperature as high as 60 degrees Celsius. It is stable at high temperatures, it does not denature at 60 degrees Celsius. So optimum temperature is near 60 or just a little less than 60, so it's still working till it's 60 degrees Celsius. Stable at high temperature. Now, enzyme 2 is found naturally in many bacteria. Enzymes for use in washing powders are obtained from bacteria. Describe how bacteria are used to produce enzymes for washing powders. How bacteria are used to produce enzymes? The main thing is bacteria grown in fermenters. They provided food substances like glucose and minerals and food, or you can say amino acids, or you can say culture medium, and then oxygen is provided, and the bacteria grow and reproduce, increase in number. And these enzymes release enzymes, and then the enzymes are separated from the bacteria, and the optimum conditions are maintained either at 26 degrees Celsius or a pH between 5 and 6. Coming to the last part of the question, pectinase is an enzyme that breaks down compounds known as pectins. Cell walls. Cell wall, please remember cell wall. It's not the fruit wall. Cell walls of fruits. So fruits are made up of plant cells and the walls are made up of, such as apples and mangoes, contain pectins. Explain the advantages of using pectinase in fruit juice production. You see, if the cell wall is broken down, then more of the juice will be extracted. So extracts more juice, or you can say speeds up juice extraction. The pectin is converted to sugars, so juice is sweeter. Just like cell wall is made of cellulose, and cellulose is actually glucose joined together. So the juice will be sweeter. Cell wall material is removed from the juice. The pectin is digested to soluble products. So the juice is clearer, meaning there are no particles of the cell wall, the pieces of pectin in it. So this is what we had to understand about pectinase. This is of course added to the new component, so that's why I've found these questions on this. So the advantage of using pectinase in fruit juice production will be why? Because it is going to extract more juice. Why? Because the pectin is converted to sugars and the cell wall material is removed from the juice. So the cell wall material is removed from the juice, or you can say the pectin is digested to soluble products. The cell wall material is removed from the juice. So the juice is clearer. Clearer means you can't see any particles on it. Your light passes through it, clearer. So this is of course very typical biological English words that we have to use, and some of you need to memorize this if you don't know these terminologies. So that finishes this video on it. I will continue some more questions which will be completed in the video following this. And thank you once again.